What's up, Bio Penguins? Today we do 2016 number 6 on eDNA and experimental design. Living and dead organisms continuously shed DNA fragments known as eDNA into the environment. To detect eDNA fragments in the environment, the polymerase chain reaction can be used to amplify specific eDNA fragments. eDNA fragments of different lengths persist in the environment for varying amounts of time before becoming undetectable. As you see in figure 1, these short fragments are going to stay around a longer period of time than the long fragments. Long fragments are a very short time. So to investigate whether silver carp, an invasive fish, have moved into from a nearby river system into Lake Michigan, researchers tested water samples for the presence of eDNA specific to silver carp. So here we're seeing the Lake Michigan, and we can see that there's no silver carp there. We can see the river systems, and we can see that there is silver carp in those river systems. So justify the use of eDNA sampling as an appropriate technique for detecting the presence of silver carp in an environment where many fish species are found. So thinking about trying to collect samples. It's a little bit easier to collect a water sample and test for eDNA than it is to actually look for the fish or identify that fish. So eDNA allows for detection of the fish without visual identification or catching of the fish. The second question is propose one advantage of identifying long fragments as opposed to short fragments for detecting silver carp. So the biggest one is let's look at the data table. A long fragment is going to only be there for a shorter period of time, which means that if I can find the long fragment, that means that that silver carp was there in a more recent time. Also, having a longer fragment allows you to be able to look for specific sequences that are specific to that silver carp. A longer fragment allows for more of those, as well as a base pairing that can increase accuracy, specificity, or confidence that it is actually silver carp and not something that's related. So student says eDNA is a appropriate technique to identify the presence of silver carp because through PCR, scientists can contribute different lengths of DNA of different species of fish, including dead ones. So while one may not physically see silver carp, the identification of its eDNA will provide evidence that the carp had moved in. Second thing, propose one advantage of identifying long eDNA fragments as opposed to the short ones. So the advantage of identifying long fragments is that the eDNA may have been shed more recently or within 2.5 days. Finding long eDNA can help scientists estimate how many silver carp there are in that area. So part B, the researchers tested a large number of water samples from Lake Michigan and found the eDNA specific to silver carp in a single sample in the lake, as indicated in figure 2. The researchers concluded that the single positive sample was a false positive and that no silver carp had entered Lake Michigan. Provide reasoning that other than human error to support the researchers' claim. So we need to say, well, how can I have gotten eDNA in the area when there wasn't an eDNA? So the easiest way is just thinking about the water. If the eDNA is in that water, then anywhere that water goes can show that the fish was there, even if the fish wasn't there. So the river flow, boats moving in the area, if an individual has so some type of organism has eaten the fish, maybe part of their wastes could have had that eDNA in them. So any of those could have been a way that it got to the lake. So the sample could be a false positive because water from the streams that contained silver carp could carry their eDNA into the lake. Hope that was helpful. Remember, AP Bio Penguins, just assess bio.